Alright, so in this section we're going to take a look at some neurotransmitters and then from there move on to a few more topics as we start to approach some bigger scale processes going on here with the nervous system. Acetylcholine is the most, familiar of the neurotransmitters that we've talked about so far, just because we talked about it quite a bit when we discussed the muscle contraction story. Acetylcholine, of course, is the neurotransmitter which works across the neuromuscular junction, or the synapse between a neuron and a muscle cell. Acetylcholine then crosses that space, that synapse between the two cells, and results in that muscle cell contracting. So again, this is a neuromuscular junction. Acetylcholine bonds to that postsynaptic cell's membrane. It's going to cause its own chemical or ligand-gated channels to open up. This is now in the muscle cell, of course. And that results in formation of what's called an EPSP, or excitatory postsynaptic potential. This is going to be a situation where the EPSP is going to cause that next cell in line to have its own depolarization event to the point where it may even approach or exceed its own threshold, at which point it will then, in the case of a muscle cell, contract. So acetylcholine is excitatory. The more acetylcholine that's there, the more depolarization we see of that postsynaptic muscle cell, and the more likely it is to fire. Now, acetylcholinesterase, which we also talked about back when we covered our muscle contraction story, is the way we're going to break down the acetylcholine once it's time for that muscle cell to relax. So again, we don't want that acetylcholine staying in the synapse any longer than absolutely necessary. And acetylcholinesterase is going to allow us to break down that acetylcholine and get it out of the synapse so that the muscle cell can relax. Glutamate is a neurotransmitter that is amino acid based, and we see this one actually more so in the central nervous system. So dealing with interneurons and their communications and interactions with one another, they are going to use glutamate as an excitatory, stimulatory neurotransmitter to cause other cells in line to have their own depolarization events. Glycine and GABA are both inhibitory neurotransmitters. They are going to have an antagonistic effect to glutamate in the central nervous system, so they kind of go the other way. They're going to cause more of a hyperpolarization and move any associated cells further away from that threshold, further away from achieving their own action potential. The way they do this is by opening up the chloride-gated channels, which to this point we really haven't talked about too much, but chloride-gated channels are going to open and allow for a further hyperpolarization of the cell in question. Because they're negatively charged, if they come rushing into a target cell, that's going to cause the inside of that target cell to become even more negatively charged. And of course, if that's the case, it's moving it further away from that threshold where it might actually achieve an action potential. So glycine and GABA are both going to result in the formation of what are called IPSPs, or inhibitory postsynaptic potentials. They are hyperpolarizing the cell and making it therefore less likely to fire. So in this case, this would be the situation where we would see something like glutamate, for example, in the central nervous system. Glutamate, remember, is the one that's going to result in EPSPs in the central nervous system. So with glutamate, this neurotransmitter hits a cell in a receptor in the postsynaptic cell. The neurotransmitter hits the receptor in the postsynaptic cell, and that triggers these sodium ions to come rushing into the cell. Because sodiums are entering the cell, because sodium ions carry a positive charge, that's going to cause this little bump in the potential across this postsynaptic cell's membrane, causing it to become a little bit closer to the threshold and just a little bit more likely to fire. Of course, if we have even more of this occurring, this little bump could become even more pronounced, and at that point maybe even approach that threshold, allowing for the cell to achieve its action potential. On the other hand, glycine and GABA are going to both have the effect of causing the cells to hyperpolarize. So if we take this neurotransmitter, the purple triangles, and plug those into the receptor in the postsynaptic cell, those purple triangles allow for this transport protein to open up, and that's going to allow for chloride ions to come flooding into the cell. As those negatively charged chloride ions come flooding into the cell, the inside of the cell becomes even more negatively charged. That takes us further down below negative 70, maybe it's negative 72, negative 73, whatever the case, it's moving us further away from threshold and therefore now making the cell less likely to fire. Some other neurotransmitters, ones that we call biogenic amines, are epinephrine and norepinephrine. So both of those two are involved in the so-called fight or flight response, which we've talked about periodically throughout the course. Norepinephrine in particular is a neurotransmitter, whereas epinephrine can be But it can be a neurotransmitter. It can also be a hormone, as we're going to see in our endocrine unit. Dopamine is important in parts of the body that control body movements, parts of the brain that control body movements. Dopamine's also involved in other aspects of regulating mood and so forth. Reward centers of the brain and so forth are stimulated by dopamine. And serotonin is involved in sleep. It's also, of course, sort of famous for being implicated a lot when it comes to depression. Insufficient serotonin levels have been implicated to cause depression in various ways. So that can be important for a number of reasons as well. We have a few others. Substance P is one that's activated by sensory neurons in response to painful stimuli. That one's a neuropeptide, as are enkephalins and endorphins. Enkephalins and endorphins are antagonistic. They work kind of in opposition to each other. Enkephalins tend to make a painful response seem even more painful, so they will intensify the intensity of the pain perceived, versus endorphins, which tend to kind of mediate that perception of pain. So endorphins are going to cause you to not feel pain that's being experienced by the body, versus enkephalins, which are going to make you feel like the pain is even more intense. Endorphins are sort of part of what's called a runner's high sometimes, or endorphins are what might cause a football player or a soldier, if they're injured, to keep on playing or keep on fighting and be able to ignore pain, even though pain might be fairly severe. Endorphins released in the brain can cause you to sort of ignore that pain, and it's almost like it's not even there if that's the case. Nitric oxide, which we also saw back in our circulatory chapter, is involved as a neurotransmitter as well. It's going to allow for smooth muscle relaxation, which, as we talked about before, is what allows for vasodilation of arteries and arterioles in the body, thereby allowing more blood to flow more freely. This one, nitric oxide, is actually a derivative of the amino acid arginine. So nitric oxide is given off as a byproduct of the breakdown of arginine, which is kind of an interesting sort of relationship there. When it comes to the actual functioning of neurons across a synapse, the way we've described this so far is by having a single neuron or a single presynaptic cell communicating with a single postsynaptic cell. Maybe it's a muscle cell, maybe it's a gland cell, maybe it's another neuron, whatever the case. In reality, that is a very overly simplistic way to sort of consider how these synapses are actually set up. What is more likely is what's happening in this picture here, where you have a single neuron, kind of the focal point of this picture, and it is receiving stimuli from several other axons of several other neurons. So in this particular case, this neuron is receiving inhibitory signals from two axons and stimulatory signals from two axons, or excitatory signals from two axons. So this is all a bit of a balancing act where, depending on if there are more EPSPs or IPSPs, that will eventually determine how that postsynaptic cell is going to respond. If the balance is in favor of EPSPs, that might make the postsynaptic cell more likely to fire, contract, release a hormone, whatever the case, maybe have its own action potential. If there are more IPSPs, however, that would then inhibit the cell from responding to the cells that come before, the presynaptic cells that are trying to communicate with it. So it's, like I said, it's a bit of a balancing act where EPSPs and IPSPs can sort of cancel each other out, and whichever one of those two there is more of, that's the direction the postsynaptic cell is going to lean as far as whether it's going to fire or not. Again, looking at synaptic integration, this is kind of similar to something we talked about before a bit, but if we again sort of think about this focal neuron here, and maybe pretend this is a association neuron or interneuron somewhere in the body, maybe it's in the central nervous system. Maybe there are sensory neurons feeding in from different parts of the body to this one individual interneuron. And if those cells are spread out over a sort of a big space, and they're all sending the same signal, if they're all producing EPSPs, then that association neuron is going to perceive that as being a fairly substantial stimulus, because you've got sensory neurons from a fairly wide range of the body that are all firing in a stimulatory way that are going to tell that neuron to go ahead and respond accordingly. The other way to do this is by having temporal summation. So maybe it's not just, 4 cells communicating with this one neuron. Maybe it's one cell communicating with that one neuron. But if that one cell communicating with this one neuron is firing over and over and over very, very quickly, very frequently, that could also be perceived as an EPSP by the postsynaptic cell. Because remember, those graded potentials can add up. And if that presynaptic cell is firing fast enough, that's going to create a bigger and bigger and bigger depolarization to the point where the postsynaptic cell can actually fire and respond accordingly by that one cell on the presynaptic side of the synapse. It's firing very, very fast. We're going to take a little bit of a diversion here and talk about some sort of illicit substances and how they can also affect how the nervous system functions. And the first thing I wanted to talk about is a phenomenon called habituation. So habituation is something that our bodies do naturally to all kinds of stimuli, and you've experienced this. I guarantee you've experienced this before. If you walk into a house and somebody is cooking something that smells really, really good, at first you notice it, maybe you comment on it, you say, gee, something really smells great, maybe it's Thanksgiving time, and everything is sort of wafting out of the kitchen and it smells just amazing, and you sort of really appreciate how that is, how it's going to taste at some point soon in the future. But after 5 or 10 minutes, you don't really notice that smell anymore. It sort of goes away, so to speak. And in reality, the smell is still there, but our brains are sort of tuning out the signal. Another situation where this occurs is if you walk into a room and sit down in a chair, when you first sit down, you can feel that chair pushing up on your butt. But after a couple of minutes, after 4 or 5 minutes, your body starts to ignore that sensation and sort of tune it out so you can focus on maybe more important things, like a professor lecturing, hypothetically. It's habituation, right? This is in fact, it's actually seen as a very primitive sort of learning because other animals can also become habituated against stimuli as well. This is not a real high level, sort of conscious level of learning, but it is a type of learning. Now, in terms of chemical responses to drugs, we can become habituated against all kinds of chemical substances. Typically, the way this works is that if neurons are exposed to a chemical stimulus repeatedly, many times over in a long period of time, they will start to have fewer receptors for that particular substance. Initially, when the substance is first in the body, it's got a really, really big response because the receptors have never seen it before. They're all firing, they're all responding to the stimulus, and it's a really, really large magnitude effect. But if you continue to use the substance over long, long periods of time, it begins to have less of an effect. And so to achieve the same effect the first time you took that particular substance, now you got to take more of it. And of course, when you start dealing with things like opioids and so forth that are very, very dangerous, the more of that you take, the more you're at risk of having some really, really nasty effects of those particular drugs. So this is habituation. Your cells are turning down the signal of the chemical because they've seen it so many times, and the way they do that is by decreasing the number of receptors they have for that chemical. So therefore, if you want to get the same effect, you got to get more chemical in your system to stimulate those few receptors that you have left. All right, so next up, we're going to take a look at how cocaine works on the body. All right, so what's going to happen here is that cocaine is going to stimulate what's called the pleasure pathway of the body, which is part of the limbic system. Limbic system we'll talk about a bit more later on, but essentially it's a sort of combination of structures in the central nervous system. The amygdala, the hypothalamus, and so forth are all involved in the limbic system, hippocampus as well. And so what cocaine does is that when it's in your system, it is going to prevent neurons from uptaking dopamine that they have released into the synapses. Dopamine, in addition to helping you to control body movements, is also involved in making you feel pleasure, contentment, happiness, those kinds of things. It's involved in the pleasure pathways of the body. So if you were to take dopamine, release it into a synapse, that would change your mood. And if it stays in the synapse, your mood is going to stay elevated for as long as that dopamine is in the synapse. If dopamine goes away, then you sort of lose the effect of that pleasure pathway firing substance in your body. So this is actually a pretty good graphic that shows this. In this picture up here, we've got a presynaptic cell. It's got vesicles that are full of dopamine here, and it's going to fire. The action potential travels down the axon to the terminus. Calcium ions come rushing in. Dopamine goes flooding out into the synapse, and this dopamine is going to start to fire this postsynaptic cell by bonding to the receptors in that postsynaptic cell. So this response here is allowing the postsynaptic cell to perceive the effects of dopamine. Now, normally what would happen in a situation where cocaine was not present is that dopamine would then be taken back up into the presynaptic cell, it would go away, and the effect would be over. But what cocaine does is it blocks the reuptake proteins for dopamine, so that when dopamine is released into the synapse, it stays in the synapse because it has a hard time getting back up into that presynaptic cell. These channels are all blocked, essentially, and dopamine can't get back into the cell anymore. So it stays in the synapse, it allows this postsynaptic cell to fire over and over and over and over again over a prolonged period of time. The overall effect of that, if this is happening large scale throughout the limbic system, is going to make you feel really happy for as long as dopamine is there in that synapse. But again, what's going to happen over time is that in this case, what your body's going to do is actually start producing more dopamine reuptake proteins, so that even though cocaine's in the system and dopamine's been released, if there are more reuptake proteins, the dopamine can come back out of the synapse, and that's going to take away that pleasurable feeling. So whereas the first time you tried the substance, you got a fairly big effect, fairly substantial effect as a result of that substance being in your system, if you were to continue to use the substance over a long period of time, that same amount of cocaine would not have the same effect because you're getting dopamine out of the synapse faster due to the presence of more reuptake proteins in that presynaptic cell's membrane. Nicotine is a completely different story. Nicotine is interesting for a lot of reasons. For one, nicotine actually serves as a almost like a barbiturate effect at low doses, but at higher doses, it's going to actually have a stimulatory effect. So the effect of the chemical actually differs depending on if you're dealing with a large dose or a small dose of the nicotine. Now, nicotine is also interesting based on what exactly it is and where it comes from. So nicotine is actually a plant defensive chemical. Tobacco plants produce it, and it's a very, very toxic substance that deters herbivores from feeding on those tobacco plants. If an insect feeds on a tobacco plant, even a small dose of nicotine will cause that insect to die rather quickly because nicotine is a very, very toxic substance. As it turns out, through an accident of biology, acetylcholine, which is, of course, our big neurotransmitter when it comes to muscle contraction across neuromuscular junctions, has a similar enough shape to nicotine that both chemicals will bond to the same receptors in their postsynaptic cells. So acetylcholine and nicotine are virtually indistinguishable as far as your postsynaptic cells, your muscle cells, are concerned. So how does this relate back to habituation? Well, again, if you take and expose your body to nicotine the first time, it's going to have a relatively large effect because your body's never seen it before. Your body essentially thinks that there's a big dose of acetylcholine dumped into the synapses and it's going to have a stimulatory effect. Over the long term, several decades later, if you're continuing to expose yourself to nicotine, those postsynaptic cells are going to have fewer receptors. The strength of the signal is turned down, so to speak, because receptors for both acetylcholine and nicotine, which again, as far as your body's concerned, are the same thing, there are fewer receptors for them. And if there are fewer receptors and you want to get the same effect of acetylcholine or nicotine or both, what do you have to do? You got to get more nicotine in your system, right? So now you start smoking more and more and more. You chew more and more and more to get more nicotine in your system to have the effect that you had initially the first time you first tried that cigarette or that chew or whatever it was. Over time, you've got fewer receptors, which means you need more nicotine to get the same effect. It also means that quitting smoking is one of the hardest things you can possibly ever expect to do because you've actually changed your cells at a cellular level, at a molecular level, by reducing the number of proteins in the plasma membranes of those target cells. And if you take away the nicotine and all you have left is your normal acetylcholine, your cells are not going to respond to it anywhere near as well as they should. And that's where you start getting into the cravings and withdrawal symptoms of nicotine.